right, so I will bring in the next panelists. I think we will have one video that will be shown, and then Filippo is here, so I will just bring him in, or Meryl already did it. Thank you so much, Meryl. All right, so this is the next hour, and we have two presenters now. We have Maria Börbic, which is from the Swiss Federal Institute of Technology in Lausanne, Switzerland. She could not join us today, but she shared a video with us. She is also the lead author on the Stellar paper that some of you might remember as the one that segments and annotates codex data in a very efficient way. And she co-authored that paper together with John Hickey and Jura Levkovich, which we'll present next in the next hour at 10 p.m. So it all comes together yet now in the methods section. And then it's my true pleasure to have Filipi Silva here also. He is a researcher, data scientist, and developer with expertise in dynamic complex networks, machine learning, social media analysis, and applied network science. So this is really where our team comes from, the study of networks with scientific means. He has a computational physics PhD and a very strong interdisciplinary focus. So we have worked together on mapping science papers, and now he's working with YY and others on social media networks, and here we are with biomedical networks. So I think it's interesting how these physicists are able to bring all their tools to many different sciences. And on his website, you can see that he's modeling the world through networks, and that's really near and dear to my heart, because I believe ultimately the human body is a network of networks. There are lots of networks which are actively working right now as I'm sitting here statically, but I think standing is not falling, right? It's all a very dynamic system. And so understanding these networks better across scales, the multiscale human, is really core to this all. And networks are used in the single cell world in terms of trajectories, in terms of neighborhood and co-expression pattern matrices, but they are not explicitly rendered like what I see on your background. I very rarely get to see these networks, especially in multi-scale networks. And so it's truly wonderful to have you here. So Meryl is going to start the first video here by Maria Börbic. And then we are going to go over to Filipi presenting. Thank you, Maria. It's really, really nice to see you here on video. I know we will see each other again at the next CIFA meeting in Geneva in May next year. So it is great to get this update, and I'm super hyper excited about Luna, because that would get us our HA pop in a spatially explicit way. Currently, we have cell type populations for different areas, volumes inside of the human body, but this is really a way to get them to be there in a spatially explicit way. So I sent you an email. Hopefully the paper gets published soon, and we would love to run it at scale over the spatial data we have. All right, Filipi, are you ready? Good to see you here.
Yes, good to see you.
And
I'm ready.
yes, you're in the midst of people that actually work in the clinical setting, right? They're seeing patients and actually are applying all that research to make them get healthier faster or have higher life quality. But we are on the computational side. So you don't have a wet lab, I don't have a wet lab. That's okay, as long as we collaborate well with those that have access to wet lab benches. And so it's really super exciting that you're here in person, and maybe just take it away, and then there will be questions.
Okay.
And you should be able to share your screen. I already introduced
Yes.
you as the man who models the world through networks. And it looks like you're going to go for a daring demo, a live demo. Good luck with that.
Let's try. Yeah. So yeah. Thanks, Katy. Thanks, Katy, for the invitation. Thanks everyone for being here. And today I'm going to talk about our visualization tools and methods to visualize basically large networks and the embeddings. this presentation is a little bit focused on science of science, but I made sure that at the end of the presentation, I'm going to showcase some early applications of all of this in the biomedical scenario. So just started this. So, but let's dive then into the visualizations, and after that, I can talk a little bit about our new like how this can be applied to biomedical science. So let me just move some stuff around and let's start. Yeah, so so basically all this AI systems, models out there, and which are basically neural networks. They can be really complex, and you don't right now we don't need to dive too much into them. We can still understand them as black boxes. So in which you have like data that comes to these black boxes, and we get an output, which could be like a classification, or some generated text, generated image, like that. So, however, if we just open it a little bit, you're going to see that most of these models are basically several layers of matrices, multiplications, complicated, that are like that. But internally, you may find a structure basically between layers that is embedding. So basically, your data comes through this pipeline, and it may reach a point in which it has a representation that is basically the communication between two layers. And these weights in these layers can be used as a vector representation of your data. We call this embeddings. It's also called sometimes as the latent representation or latent space for the data, but it can be interpreted that way as well. So we can recreate this black box in a way that it's still a black box, but it now has data, it has an output, but we can also take a look at its embedding there. So which is a vector representation of your data. And there are many methods that work like this. So we can have, for instance, documents, which can be images, can be um, sound, music, or text, networks, graphs. It can be anything, and then can be transformed into these embeddings. So you can have, for instance, we can use doc2vec, or transform doc2vec creates a representation for each document, or transformers that can create a representation for any piece of text. And Node2vec, for instance, that can be used to create a representation for the nodes in a network. So just to give you an example, and we can then obtain basically a vector representation of the data, and this vector representation is usually have like a high dimension. So it's a little difficult to deal with that, but it's a little bit better than the document itself. And this approach, embeddings, can be used for many tasks like recommendation systems, database, search engines, and RAG, and also anomaly detection. And it's all based on the idea that documents, so if you have a document there in this space, you can get all the neighbor documents of that document, meaning that they are very similar or very close in this space. So if you have a vector representation for each of these documents or entries in your data, you can you can know the similarity between them. You can know that like which ones are closer, so which ones are very similar. So that's a way that you can understand these systems. There is another issue, like data is also complex. And for that, I'm going to use the example of science of science. We don't need to go super deep on that, but basically it's the science that study science itself, using science. And the idea is like the main questions in this field are basically like how science is evolving, how teams are formed, what are the new technologies that are going to appear in the next few years, etc. So there's several. It's an interdisciplinary field. It involves several other fields. It emerged, I would say, recently from all these pieces getting together. But let's just dive into a small portion of that, which are publications. So for publications, we have we can have, for instance, several types of relationships, right? So if we just get a few of them, you're going to see that this system becomes really complicated, right? You have authors, you have institutions of these authors, you have the journals where these publications were published, you have the citations between these publications, you have the content of these publications like title, abstract, etc. So this can get, and all these connections are not trivial. They are really intricate. They can be complicated. They have several explanations, models behind this, like mechanisms behind this. So it can be quite complicated. At the same time, this is a field in which we need to be good at the communicating the findings or anything that we end up understanding about these systems. In particular, of course, to experts, to other researchers in science of science, for instance, and also of course, to policymakers, because they are the people interested on that, like and other stakeholders like grant agencies, the government, etc. So we need to be able to explain and communicate all these findings to them, and they may have different backgrounds. So it can be quite difficult task, in particular looking at the complexity of some of the systems that we have here. Another thing is we of course need to always communicate to the general public. This is always needed. We always need to be to have a way to deliver this to the general public. What kind of research is being done right now? What kind of perspectives and technologies are going on? And of course, companies. Companies are always interested on which technology will be developed next, if certain technology should be adopted or not. So companies have also interest of that. And we need a universal kind of way of communicating all of this. And one way that we can do that is through visualization, in particular, for instance, through maps. And maps is a quick way that we can understand relationships, we can understand how things are distributed, and we can clearly glimpse, like get information out of maps. And most of the people can understand this map. So for instance, here just showcasing one map of international collaboration in a big science project, particularly LIGO. And here you can see like the connections here indicates like strong ties between strong collaborations between institutions. The colors indicate like light colors indicate more recent collaborations, while darker colors indicate like older collaborations. And then you can see some patterns here, like when some collaborations start to happen, etc. So this is super clear, but it would be even more interesting if you can do something like this also in the knowledge space, right? By that I mean like suppose that you have a map not of the world, but a map of science itself, so you can understand where physics is or where biology is, and we can see where these actors are, like where are the authors of these papers, where are the journals, where are all the what's happening, is this growing, is this, so if you can have a map in which we can see all of these mechanisms and all these complicated structures that I showed before, it can make things easier for us to understand what's happening, at least in science. So there's a whole literature on science maps. That's the area that basically work with this type of knowledge mapping, and and that they, of course, the main objective is getting like the best map, or the best map that can be used for a certain task. or the best map for a certain data subset of the data. And yeah, so there's a lot of literature about this. And one of just to showcase one map would be something like this. So you have each point representing maybe a paper or a location, a small, tiny location, a tiny amount of knowledge in each distributed in 2D or 3D space. And you would have borders of this knowledge, borders based on the fields distributed like this. So they kind of represent the countries or some kind of structure like this. And another aspect of that can be useful is we are really good at connecting the dots. We have been doing this for quite some time now as humans, and we are good at this, and we can find patterns. In particular, these patterns of constellations are not truly real, but what I mean is that we are always trying to find patterns, and while here they are not real, they may be real in the data itself. So one way that we can understand these complicated systems or the data that is applied used in these complicated systems is basically through a network. So how we can interpret this? So we have documents that are connected if they have a relationship with another document or another a data point. Let's call it data point. They have a data point that's connected to another data point if they have a relationship with that. Could be like a citation, could be like someone migrated from one place to another, or it could be, for instance, just the similarity, right? It's just, you remember I talked about embedding. So if you have two documents that are very close, we can connect them. This could be a way that we can kind of starting understanding what's happening, connecting the dots. So, and when we do that, we start to have networks. And just to give you some names, so we consider the data entries as nodes and the connections as links or edges. So I may switch a little bit between slides, between edge and link, but that's the same thing. So just to showcase here, so we have a few network visualizations, and you can already see that just by visualizing these networks, we can see some patterns. We can kind of start distinguishing, and they start understanding these networks, what these networks are about. The problem is that when you are analyzing networks, we start to have few problems. Not problems. I mean, some characteristics of these networks that lead to challenge that we need to overcome. So, for instance, networks can be really large scale. They can be huge. Have millions of nodes, billions of connections, right? So they can be dynamic, and of course they can be complex. We already know that many of the systems that we have in the real world, they are really complex. They don't have clear, like a super trivial connections. They are having intricate connections. So we need to overcome a few challenges with that, like a scalability. We need to be able to visualize large structures. We need to also overcome the problem of if you have something that's super intricate, you may not be able to see its structure. So you may need to kind of overcome what's called like hairballs, when we normally see these kind of structures in network visualization or even embedding visualization. Another thing is we need to be able to capture the multiscale structure, because several systems, complex systems, are organized in some hierarchical structure or some kind of a cluster kind of structure, which can be also multiscale, right? So another aspect of this is, of course, the dynamic exploration of this, how we can deal with time varying networks, for instance. And our proposal here with software that we have been developing is to attack each one of these problems with a set of a core set of features that we are working on having everything implemented, of course, but we are trying to make all of this happen and overcome this challenge. So basically, we need to, the core features are basically the layout algorithms, the rendering pipeline, and then the interactivity functions that we have there. And as I mentioned before, we have been working with developing a tool itself, like a set of a library that can be used to visualize networks. And it's basically open source web framework that can be integrated into websites, portals, etc. And it has an optimized rendering pipeline. It runs inside the browser, and it provides some level of interactivity that we have been working on, but that's the main idea. So open source web based, and we are trying to make it optimized to address all this challenge that I mentioned. So first thing that I'm going to talk super briefly is layout algorithms. So the main idea of layout algorithm is to find the position of the nodes, how you position these nodes. And classically, the way that it have been using a lot is basically a force-directed layout. But basically, what you want the idea that nodes that are connected should be closer, and the nodes that are not connected, they should be as far as possible. One way that we can approach this is through what I mentioned, force-directed layout, which is basically interaction between particles. So basically, the idea is to have interaction that is attractive if two nodes are connected, and interactions that are repulsive among all the nodes, all the pairs of nodes. And by doing that, we get this idea of making nodes that are connected closer and everything else as far as possible. Of course, we need optimizations for that. So there's a few approaches that can be used. We have our own approach. I'm not going into the details of this, but we cannot just do all-to-all relationships. We need distances and calculations. We need to do some approximations and optimizations, segmentation of the space. And then we are able then to basically obtain a quick, fast, and obtain what is scalability, at least at the level of the layout algorithm. And sorry. So if you apply this type of algorithm, we're going to see something like this. So here is a weak network. So each node is an article, and only for medicine and mathematics categories. And you're going to see basically the two categories being basically split into two communities, and everything becomes very clear. Another aspect of visualization software or visualization approach is the rendering pipeline. And for that, we are using GPU. This is, of course, not a surprise. Everyone should use GPUs now for everything, if possible. But the idea of using the GPU is what brings so much powerful features to the rendering pipeline, and while also allowing us to visualize a really huge amount of data at the same time. So we are using something called billboard rendering. Again, not going into much details, but basically you just put some billboards in front of the camera, and we render each one of these billboards using the GPU with like a drawing routines, and these drawing routines can draw anything, and we have freedom to work with any type of drawing. So for instance, we can draw spheres, perfect spheres in this case. Like you see that they're perfect, sub-pixel perfect spheres. So, and here we have 20 million, and it's completely in real time in the browser. We can run this in the browser, for instance. So it's crazy how GPU can enable all of this kind of amount of things at the same time being visualized. Another advantage of using GPU and the advanced rendering pipeline is exactly improving overcoming the hairball problem. For instance, in this case, we can, if we start from this network, if we instead use not a rendering approach, which instead of rendering the edge connections themselves, we render the edge density, we see something that looks like this, which is now something that we can try to understand with at least two communities. We can start seeing stuff on this structure. So it's just to illustrate this aspect. Another type of effects that can be used, for instance, just to illustrate types of effects that can be used for that, is like ambient occlusion that allows like 3D structures to be seen much better way, even in static images. So you can, and our brains are now almost okay in understanding what's behind and what's in the front, for instance. So depth gains a little bit of if you get something new here from this type of effect. And here's just one example that approach that it's too small to see much about this, but it's just to showcase the general idea. We can create, for instance, science maps using this type of visualization, and we can interact with them and explore. So this is for physics, for instance. Here's another example. Just I am going to jump quickly just to see how normally the interface and the rendering works. So this is a network of maybe 1 million nodes from AI research papers, and we can explore these networks. We can basically go dive into them and see how improve the layout. The layout is not really good in this version. This like older thing, but we are improving that. Okay, so I talked about networks. So what about embeddings? For visualizing embeddings, we have a very similar approach. We can use UMAP, and UMAP basically internally it just creates some kind of a network-like representation, and then it applies some it's not really force, some optimizations to this to the positions of nodes, basically to make them closer if they are like closer out in the original space, and far away if they are far away in the original space. So it uses another technique. It doesn't use force directed layout, but it has so many parallels that there's some papers that kind of connect the two. You can even switch the way that you optimize UMAP to using force directed layout, for instance. Getting back to science maps, now I can talk about this map. So basically, it's a visualization of all the papers in the MAG, the Microsoft Academic Graph. So 200 million entries. We are not currently seeing them now. It's just a sample, but the map itself was constructed using all the data. And here are the fields, etc. So they're colored by the fields from NSF areas, like major areas. And we can, if you use some effects like a GPU-based effects, we can then look at the structure of basically, sorry, we can look at a distribution of nation production, for instance. For instance, we can look at where USA is focusing more and where China is focusing more on this science map. So, and this can be done interactively. So you can just switch from one thing to another, or filter out stuff, and it automatically creates maps like this for these distributions. Another example is COVID-19. So this is another science map, and we can look here down here if we see this bright region. These are all the COVID papers, or like COVID papers, at least in your data set. And there are a few more, like I spread, but most of them are concentrated around here. And here you see where the citations of these papers. So we can try to understand what kind of things were used during COVID in terms of research. We can look, for instance, also where certain communities are. For instance, where the NetSci community, the network science communities, in terms of the conference, where they are in the science map. And here, for now on, I'm going to show a few maps that were obtained using different ways. And you're going to note that the structure, like the overall structure, is almost always the same. You have some kind of like a cycle a circle with something void in the middle. So if we enable density, we can see that's less, it's less dense in the middle. And this seems to be persistent. This was observed by Katy, by Clevens, and several other, not several, a few other outdoor researchers in house that contribute to Science of Science. And it's quite, it seems to be quite robust independently of the type of network that we use. So for instance, this is a journal network, journal to journal network. And here we have Wikipedia articles. We still see this cycle here. So, and we have been building these models for quite some time. And you see that all of them have some kind of similarity, and these are different models. Like this is just to showcase that depending on the model, we get something a little bit different, etc. But the main question that we have is basically, can we understand why, for instance, two elements are very similar or very close together in these maps, or even in the original space, and why some of them are far away? Of course, the topic is a big player here, but it's not only that. There's several aspects to that. So, but the current models doesn't allow us to answer like well these questions. We don't know why they are together. Are they because people are citing the same stuff, or people are like or the subject is exactly the same? What's the how we can do that? So we have been developing methods, in particular with YY and Sadamori Kojaku, in which the similarity between we have between different regions is based on that transition of authors. So we are forcing this, we are fine tuning these models so that we can interpret this there's different similarities, for instance, or different or distances in some sense. So if they are closer together, it means that there are many people or the same authors, they are basically acting into these two topics. But if they are far away, it means that there are not many of them, or maybe none. And by doing that, we have a map that now looks like this. But in this map now we can understand what the distance means, like what similarity means in this map. So it's a better interpretability of these complicated models. We can do some stuff with that. What I can say is that it works well for tracking the trajectories of these researchers, and it can even predict new collaborations and can also predict the emergence of new journals. And I think that I don't have much time. I'm going to just brush a little bit on this, but basically we also have been developing ways that we can improve the user interface, in particular because if you have a general visualization software, you may not have a general, it's really hard to get a general user interface, in particular because it's going to grow more and more complicated as the data as the needs are different. So our approach, or tentative approach at least, is to integrate this with LLMs, to large language models, and have like a ways that we can interact with these visualizations just by chatting or even speaking with these networks, network visualizations. And then we can map wherever we want. We can do customized kind of mapping of colors and attributes to visual attributes to real attributes. So that's a way that we can do this type of interactions without creating a huge, complicated interface. I'm going to skip just a few of these slides, but I just want to say that we have been implementing these tools for several different areas, like social science, for detection of coordination, for instance, in social media, social media networks. And also we have been adapting this now for biomedical research. In particular, I have been working in single-cell RNA-seq visualization, microbiome association between tissue and the microbiome population as well, and the tissue gene expression, some brains networks changing over time across age, and metabolites networks as well. Here are some recent works that we have been doing. So just to say that we have been this very recent one, like just published, not published. It's on bioRxiv last week, and it basically we are comparing different community detection algorithms in single cell data, and we found that visualization was, of course, super important for that. And here is just some. It's just a sneak peek. I'm not going to the details of this because it's unpublished. It's not. I just want to leave this like running while we may wait for that. Like if you have questions, I will be happy to answer. But this will be like the end. So this is a biological single cell type of network. Embedding, and then we transform this into a network, and you can visualize this huge network and interact with it and see different types of structures like cell types, the information about the patients behind this data, etc. So and this is in collaboration with Katy. It's a super recent thing. So yeah, so thanks so much. And I just want to acknowledge all these people that have been working with me on all these projects. And yeah, and that's it. Thanks so much.
Thank you very much, Filipi. I think really some of these techniques that are developed in physics, in computer science, in science of science, and other studies could really transfer over to the very UMAP dominated world of single-cell studies. And YY and Santo Fortunato and others at Indiana University really have the expertise to advance these clustering and embedding algorithms. And so it's really wonderful that you could join us here and give everybody a taste of what this future might look like. Ultimately, I would love to know what is the largest number that you can lay out. So not just showing nodes on pre-computed positions, but really do a network layout.
Yeah, so we can, if you have a strong GPU, like in a desktop machine, we can do millions of nodes. They cannot be. We still are at the sparse regime, so we cannot go to the dense regime in terms of edges. But we can do millions, like from 1 million to 10 million potentially. 10 million is a stretch of what we normally can do at the same time. But normally we use sampling techniques and even like interactive sampling, so that if you zoom in to certain regions and want to see more, it shows up more stuff as you zoom in. So that's something that can be used to reduce kind of the clutter, but at the same time improve performance. But we are targeting the region of millions of nodes. We cannot go much beyond that in a web server. For anything beyond that, I think we can do. We need clusters, technology, and all this coordination between different machines to provide to create the visualizations. It will be a challenge, like a cyber infrastructure plus physics plus like involving everyone, like I mean different disciplines to get this beyond that level, I think.
Yeah, and I think Aviv Rajev earlier today mentioned 100 million cells for RNA-seq data that's now available publicly.
Okay.
And for the spatial data, we are at 50 million within HubMap++. And so I think spatial data will get bigger quickly.
Yeah.
And I think then it's a matter of intelligent sampling, right? There are some very, very rare cell types that you don't want to lose, but then there are also many cells that are very abundant everywhere. Yeah, so I'm looking forward to discuss more. We have reached our 10 o'clock time here. If you could put the link to the new paper papers that just became available on bioarchives last week into the
Yes.
chat before you leave here, because then you don't have abilities any longer. That would be great.